Okay, I think we can make a start. Thank you everyone for joining us today. Really excited to provide you this overview of our next generation breath analysis towards accelerating non-invasive biomarker research. My name is Hugh Davies. I'm heading up our business development team here at Owlstone for our research products and services. I'm going to give an introduction to the company and also cover some of the technology, the different technologies we have for breath biomarker research. And then I'm going to pass on to Tilly, who's going to go into more of a deep dive on the latest platform that we've developed here really for robust breath research. So Tilly's our lead analytical R&D scientist here at Owlstone Medical. Just to say, in terms of housekeeping items, please put questions in the Q&A box. And then for anything else, you can use the chat function, and we will come back to those questions at the end, and myself and Tilly will address them at that point. So with that, I'll get started with the presentation. So just to give you a little bit of background on Owlstone as a company, which might be new to many of you. We're based in Cambridge in the UK, and we formed as Owlstone Inc., which was a spin out from the university back in 2004. And then in 2016, we spun out Owlstone Medical. So the original business was focused more on military and industrial applications of volatiles in the environment. Owlstone Medical is very much focused on medical applications around breath analysis and other biological sample types. We're actually close to around 200 people headquartered in Cambridge, and we've got over 15 years experience working with volatile molecules across a wide range of different industries, as I mentioned. Very well capitalized, lots of patents covering the technology. And our breath biopsy technology is used in over 200 research labs and clinics around the world. Good body of publications around that. We're running some of the largest breath-based clinical trials as well. Just to give you a little bit more detail on the business, our end goal really is to develop breath testing in every home for everyone. Breath testing is obviously non-invasive. It can be done at any time, any place, and can be used for monitoring of multiple diseases potentially. And really, our goal here is to develop breath tests directly and in partnership with academic, pharma, and diagnostic partners. And we can break the business really into three main component parts. We have our own internal investment into test development, both for biomarker discovery and validation, and then ultimately the development of those tests, primarily in the areas of early detection of lung cancer and then precision medicine in digestive diseases such as liver disease and gastrointestinal diseases like IBD and IBS. We then offer up this platform technology that we're going to talk through today for research products and services customers in the academia as well as pharmaceutical companies with active studies across a wide range of different disease areas. And then the last part of the business is OMED Health, which is basically where we're developing at-home tests and point-of-care devices for released under the OMED Health brand that we ultimately would look to either develop ourselves or out-license to pharma or diagnostic partners. And the first area there is in gastrointestinal disease that we'll talk about more in a moment. So first of all, why breath? For those of you new to the field, I think most obviously it's completely non-invasive, pain-free, and well tolerated by patients. It's an inexhaustible supply, of course. You can always breathe more, unlike other tissues and biofluids that might be used as biomarker matrices. These tend to be volatile metabolites, so they can be reflective of what's actually happening phenotypically. And through the processes that we use today, we can accurately chemically identify those molecules as well and link them to biology and mechanistic associations in the same way you can with other types of omics approaches. Again, with the modern mass spectrometry and techniques we've developed, we can get down to very low levels of limits of detection, down to parts per trillion in some cases on breath. through pre-concentration and high sensitivity mass spectrometry. So really seeing some of those more biologically relevant molecules on breath. And then because it's non-invasive and available in an inexhaustible supply, of course, it's compatible with high-frequency serial sampling, for example, post-treatment with a drug or nutritional intervention or food. And it's not just for respiratory disease. Yes, breath, of course, is proximal to what's going on in the airways. And a lot of the work we do are in respiratory diseases like asthma and COPD. But those VOCs can also perfuse from other organs and tissues in the body into the blood and then onto the airways. So it's really more of a total body snapshot of biology on breath. And of course, it's an orthogonal approach to other biomarker platforms, so it can provide additional data to, for example, genomics and proteomics experiments. And then finally, and we'll touch on this more as we go through the presentation, because it's breath, it's has the potential to translate to point of care or home-based testing in the future through, for example, sensor-based devices. So our focus as a business is very much on the gas phase of breath or exhaled VOCs. And human breath contains a very wide range of VOCs that can originate from both endogenous as well as exogenous sources. And they can represent metabolism throughout the body, as I said before, originating from both the airways as well as from other tissues and carried through the bloodstream onto the breath. When we think about the type of biology that we can study in breath, it covers a wide range, but this slide kind of talks through some of that. So in terms of endogenous metabolites, these can be molecules that are influenced by inflammatory processes, in particular reactive oxygen species impacting on lipids, and through lipid peroxidation lead to the release of molecules such as aldehydes, and then subsequently alcohols and alkanes. So these are well represented. Fatty acid oxidation is another area. So molecules such as ketones, which are implicated in metabolic diseases like diabetes, molecules like acetone, 3-hydroxybutyrate, etc. And then we also have a very good representation of bioactive molecules coming from the gut microbiome itself. So things like short chain fatty acids and products of aromatic amino acid degradation. So things like indole and 2,3-butadiene, etc. So this is a very interesting category of molecule for us and for the field in general. And then volatiles, of course, can be from exogenous sources, so related to exposure to smoking, pollution, workplaces. So volatiles that we're inhaling but remain within the body and then are exhaled at some point in the future, as well as dietary components such as terpenes and other volatile molecules from the diet, as well as drugs and drug products. So exogenous compounds are interesting because they could be involved in disease initiation and disease pathogenesis when you think about, for example, pollution and lung cancer. So the VOC having a biological effect, but also how the body metabolizes those exogenous volatiles as well is interesting in terms of differential enzyme activity, for example, linked to a disease. And this has formed the basis of many of our test programs, particularly lung cancer and liver disease, where we're taking exogenous VOC probes that can either be inhaled or taken orally, are then metabolized by enzymes in the body, and then release a volatile reporter back on the breath. So that can become a way to probe a very specific piece of biology. So there are some challenges with breath and VOC analysis. It's not a new field. You'll see lots of papers out there with people using different approaches to both collect and analyze breath. Obviously, everybody breathes differently. VOCs are present in environmental air that I touched on that people inhale, another source of variability there. There's a very diverse range of concentrations and chemistries of VOC, so you need powerful analytical technology to analyze all of those. So a lot of our efforts within Owlstone has been to develop collection and analysis tools that allow us to address some of these challenges to really minimize technical variability, minimize the noise and background that can come from the air around where the sample's being taken, or indeed the collection equipment itself, and then to be able to be analytically very broad in terms of the range of VOCs we can measure, and then also make sure that we can accurately identify what those volatiles are as well, because that's obviously an essential part of this in terms of understanding their biological associations, but also in terms of following them up in validation studies and ultimately creating a test. So some of the core technology we've developed is wrapped into this breath biopsy collection station. There's different elements to this. There's the receiver breath sampler for robust breath sampling captured onto sorbent tubes within the device. There's the Casper portable air supply, which is about the size of a desktop, small desktop printer, which filters the background air in the room, the ambient air, scrubs the VOCs from that air and provides basically filtered line of air into the receiver. And then the Breath Biopsy Collect software and a laptop that allows for metadata collection, but also monitors the breath collection during collection. So the receiver breath sampler itself, as you can see, has got a mouthpiece interface. It's a disposable item. The patient breathes into the receiver. It's just normal tidal breathing. There's also a nose clip to stop them breathing through their nose because they're breathing that filtered air from the Casper. So I said it's normal tidal breathing. Within that device, you've got CO2 and pressure sensors, which allows the software basically to learn the patient's breathing profile and then activate pumps to collect a specific fraction of each breath and draw that breath and the volatiles from that breath down onto the sorbent tubes, which basically allows for a pre-concentration of the volatiles onto the sorbent tube over time. We collect a fixed volume, and then once the collector's completed, generally takes about 10 minutes, the sorbent tube is then released from the device, end-capped, and then shipped back to our lab for analysis. So if we just look at some of this in a little bit more detail and some of the advantages of doing it this way, it does allow for a pre-concentration of volatiles from multiple breaths. That really allows us to enrich for those lower abundance, more biologically relevant VOCs from a breath sample, and really allows us to get a very wide range of molecules to analyze. It minimizes issues associated with background environmental contamination. So the Casper portable air supply basically scrubs those VOCs from the air that's breathed by the subject providing the sample. It's a very precise collection of VOCs. So we're collecting a specific volume of breath. Typically, we collect 5 liters of breath across the 4 tubes within that cartridge. And then we're also enriching for a particular breath fraction. So we're generally fractionating on the end tidal or alveolar fraction of breath and eliminating the first part of the breath, including the oral cavity, which tends to be highly variable and can dilute the signal. The computer-guided collection allows us to minimize differences between site staff. And then finally, that device is also built from inert materials, which can minimize leaching of VOCs into the sample, which all can create, false positives and false negatives when you're doing biomarker research. So once that sample has been collected onto the sorbent tubes, I said that can just be end-capped, shipped at normal room temperature, back to our lab in Cambridge, where we can perform high-resolution, accurate mass GCMS analysis. That's thermal desorption GCMS we're using on Thermo Scientific Q-Exactive Orbitrap instruments, which are highly sensitive, high resolution, allows to profile a very wide range of volatiles from the breath sample in a very accurate way. Once we've collected that raw data, it can be subjected to deconvolution, feature extraction, normalization. And then on the back end, we can match the data against our VOC library. So it's a high resolution accurate mass library of chemical standards that we can match against the experimental data to have high confidence identification of the VOCs, as well as search against publicly available libraries like NIST, where we don't have a match to our library. And then on the back end of that, we can do statistical analysis and interpretation of the data. So we have a team of metabolomic scientists on staff that can provide a report in terms of a PowerPoint presentation, a biological interpretation, and suggest putative biomarkers that have come from such a discovery study. We're not limited to just looking at human breath. We've also devised ways to look at mice samples, as well as in vitro and other types of biological samples. The mouse method we developed together with the pharmaceutical company Boehringer Ingelheim. It's a flex event model where we can collect breath from intubated mice, a very clean sample. And then the in vitro capability, we're using so-called high sort probes and automated robotic handling of the samples. And that allows us to look at cell culture media and other biofluids like urine, serum, stool samples, etc. So it gives us a flexibility to work both preclinically as well as matching that up with what we're seeing on human breath as well. And the back-end workflow is largely along the lines of what I just described in terms of the mass spec analysis, and Tilly will be going into that a lot more detail. The final point to this, and again, Tilly will be covering this, so I won't spend too much time on it here, is that we're also developing a VOC breath atlas. So that's a growing list of VOCs that we've shown are truly on breath across different studies. So we can look at baseline levels of VOCs, for example, in healthy control individuals, as well as how those VOCs may differ in different disease states. So that's a really useful resource that we're building up that helps with validation of VOC biomarkers. One of the other aspects of what we're doing is really sort of an end-to-end process. So I've talked through a lot of the discovery capabilities that we have, but we also have capabilities in the business to create those tests on the back end that can be used clinically. And that could be, a lab-based test, or it could be some simple home-based collection method that allows for a collection of breath samples remotely or in the home, with those samples then coming back to a central lab for analysis with mass spectrometry. Or it could even be, a handheld device that allows for both the collection and analysis in real time. And I'll talk about some examples of where we've done that previously. So this is a kit-based business that we've launched in the UK. We've analysed over 100,000 breath samples so far clinically. This is designed to measure hydrogen and methane gases on breath with a carbohydrate substrate that's taken by the patient. So that'd be a molecule like glucose or lactulose. And then samples are taken before and after the carbohydrate substrate for diagnosis of common gastrointestinal conditions like small intestinal bacterial overgrowth, intestinal methanogen overgrowth, or carbohydrate intolerance. So this kit we've devised, though, it can be just sent off to the patient's home. They provide the breath samples, which then comes back to our lab where we do the mass spec analysis. And we're working with the functional gut clinic in London, who then interface with the patient to make that diagnosis and put the patient onto a treatment plan. Another example, and this is kind of the next iteration of what I've just described, is an at-home real-time analysis of hydrogen and methane. You can see the device there. It's a small footprint device, low cost. And this allows for basically testing of multiple substrates and longitudinal treatment monitoring, for example, but also allows for sort of quick, convenient data collection, logging of food, drink, symptoms, lifestyle records, etc., within an app that we've also designed here at Owlstone. So that allows you to track progress and daily data collection. It allows you to do data overlays to spot trends and correlations between the data you're entering and the real-time levels of hydrogen and methane that have been measured on the breath. And then the other component to this is it also stores records automatically in a secure centralized database that can be shared with the treating clinician as well, in terms of resolving conditions related to IBS that I just described. So the reason I'm mentioning this is that it's a demonstration that we have these capabilities in the business, not just on the discovery side, but also to develop this type of sensor technology for the future as well, non-biomarkers that we've discovered. And this is one example project where we've done this with the US Department of Defense. This is the Defense Innovation Unit, part of their Exhale project, which is very much focused on the early detection of human infection using VOCs on breath, with the end goal of developing a handheld, ideally handheld, fieldable device for non-invasive detection of pre-symptomatic respiratory infectious diseases out in the field for military personnel. So very excited about this project. It's in flight at the moment. We've done a literature survey of potential biomarkers that are already out there. We're also running clinical trials ourselves through Duke University in the US to identify new biomarkers of viral and bacterial infection. And then ultimately, once we've defined a panel, that will then be moved into a fieldable device like I've just described. And we have other projects in the work that are similar to this. So I'm now going to hand over to Tilly, who's going to go through in a bit more detail on the VOC atlas that I talked about, as well as talk through some of the advancements that we've made in terms of the platform technology over the last year or two. Thanks, Tilly.
Thanks, Hugh. Thanks. So we've already set the scene for the customer study workflow using the receiver and the breath biopsy platform, which is what we use to collect patient breath samples. So I'm now going to go into more detail on the analysis method itself and the breath VOC atlas and how they kind of aid us to deliver customer studies. So our breath biopsy atlas currently compiles of over 200 verified volatile organic compounds that have been found on breath in a diverse group of patients using our breath biopsy OMNI sampling platform and analytical technology. So this list undergoes continual expansion with compounds added from customer studies and literature reviews in disease areas of most interest to our customers. So the breath VOC atlas provides multiple benefits for us. It deepens our understanding of the biological pathways associated with breath VOCs. It enhances our grasp of their behavior in various populations, and this can lead to improved study designs. Improved study designs and enhancing and kind of making changes to our sampling platform, such as breath collection, sample storage, analysis, and feature extraction. It's a reference data set that can be drawn from for future studies and therefore can be used for in silico comparisons between studies to help us accelerate biomarker development. And as part of this continuous growth, new customer studies that are run within Owlstone will feed into Atlas to expand its overall offering. So what are the key building blocks required to create an atlas of VOC compounds on breath? And I'll explain the significance of the on breath label in the next slide. So firstly, a sample collection platform, which is well established within Owlstone, and Hugh's already alluded to this. Adding to that robust sampling, we collect quality breath and blank study data from customers and within Owlstone. So we can now move on to the analytical platform used to analyze these samples. So combining the next generation analytical methodology and a dual capability data processing mode, this allows us to look at a breadth of targets within the targeted part of this, which has 150 compounds, but it's alongside our usual untargeted feature extraction, which is currently used within Owlstone. And the use of these is tailored to generate us the best chance of finding valuable clinical biomarkers. So why does on breath matter to us so much? Other studies out there typically quote the number of detected compounds, which can be very high, but as we've seen from experience, many of these are contaminants from the environment and don't necessarily relate to patient biology. For a VOC to be a suitable biomarker, we need to be confident that it's coming from breath and not somewhere else. The air that the person's inhaling or the sampling hardware of the boundaries, and to ensure that our library is populated with VOCs that we're confident could be good biomarkers, we're constantly mapping VOCs that we see as on breath. So just to make a quick distinction here at this point before I carry on between the two compound groups that we're talking about. So Atlas, which is our in-house library, contains 200 compounds. I mean, it's a reference library which can be used post-data processing to look up relevant significant compounds detected in a study and to cross-reference them against literature and other studies run within Atlas, within Owlstone. Adjacent to this is the targeted panel, which I'm going to talk about in regards to the analytical method. And this has over 150 compounds made up in a quantitative chemical standard preparation, which is run alongside our method and our studies as a calibration curve. This produces a targeted feature table, which is available to the customer for these specific compounds of interest, which have been validated on the method. So the targeted compounds are taken from Atlas, and we hope that more Atlas compounds could be added to the targeted panel over time, but they are two separate groups that we're currently talking about. So moving on then to our targeted plus method and how this helps us expand our on-breath Atlas. So the next gen analytical method was developed to improve our global VOC analysis platform and the operational delivery of customer studies. And we've coined it the targeted plus method as it has dual capability with over 150 targets in the targeted portion, but it's also well suited to untargeted analysis. This method is being developed to improve analytical capability, including better precision of our on-breath targets, better compound identification for features outside of the target panel, and increased comparability of data within studies. So this overall improvement gives us better quality data to be able to grow the Atlas library. Subsequent customer studies will be run on this method and the results will be fed into Atlas over time, and then hopefully those compounds will be re-added to the targeted panel, and it will grow over time. It will evolve as we complete more customer studies. So just taking a look at what we have in our Atlas. So the VOCs present in the current Atlas were investigated to determine their biological origins. 10% were related to the microbiome. 52% were associated with the human metabolism, particularly pathways involving oxidative stress, lipid peroxidation, and inflammation. And 37% were related to exposure. Among this subset, the majority stem from food sources, including flavoring agents, industrial processes, and cigarette smoke. The majority of papers discussing these compounds in relation to disease focused on respiratory diseases, cancer, and metabolic gastrointestinal diseases. And you can see these on the pie chart. And the table below the pie chart shows a few examples of some of these compounds that are listed in our Atlas. So zooming in a little bit on the microbiome, as mentioned previously, the targeted plus method has over 150 target compounds. A subset of these can be attributed through literature to have associations with the gut microbiome, which is an area of great interest to our customers. So 28% of our target panel contains compounds such as short-chain fatty acid, which draw links back to gut health and the microbiome, along with many other compounds from different classes as shown here. These are all in our targeted panel and in the Atlas, which means they benefit from quantitation, confident ID, and the ability to track across other studies within Atlas. So the volatile metabolites derived from the microbiome, which are, as seen on breath, encompass compounds like short-chain fatty acids, as I've mentioned previously. These compounds have been associated with a spectrum of disease states, spanning neurological disorders. Here they reflect the intricate processes of the gut microbiome fermentation involving sugars and protein substrates. So an important step to building the VOC Atlas is having a suitable analytical platform. The TD-GC-MS instrumentation is considered our gold standard analytical approach for breath analysis, where VOCs over a wide analytical concentration range can be collected via sorbent tubes and separated and analysed by a GC-MS. We want a method and instrument platform, that's capable of dealing with the complexities of a breath matrix, something able to cover that wide analyte concentration range that goes all the way from really trace compounds to very high abundance targets. We need the sensitivity and the high resolution accurate mass to facilitate structural elucidation of features during our discovery analysis. So I'm going to delve more into what the high resolution accurate mass aspect of the instrument brings us. But from here on, to avoid the tongue twister, I'm just going to refer to it as HRAM. So a deep dive into our targeted plus analytical method and kind of some of the benefits from both sides of the camp. So we have the targeted and the untargeted. So the targeted analytical method, as mentioned, analyzes a wide range of targets spanning various chemical classes and volatilities. And this approach enables us to have a single tube analysis. So we collect 4 tubes, but we only have to run 1, which gives us a chance to run potentially on other orthogonal methods and repeat analysis if we should like to, with high accuracy and precision. It enables us to have reliable results across the extensive targeted spectrum. And a key feature of this, as also mentioned before, is the planned iterative panel expansion, which allows us to adapt and evolve to research needs. If we want to add new compounds or remove compounds, we can do that. With the wide analyte concentration range, like I said, the method accommodates both the very high and very low abundance biomarkers, which we've seen across Owlstone before. This versatile analytical method is designed to deal with targeted and untargeted analysis within the same data set, which offers us depth in chemical characterization. The untargeted workflow leverages MS full scan mode, which enables us comprehensive profiling of complex samples, capturing a wide array of compounds without prior target selection. Additionally, the method integrates the HRAM library, which allows us high confidence compound structural identification. And combining advanced instrumentation with tailored workflows and comprehensive data analysis tools, the method represents a powerful tool for unraveling some of the complexities of the chemical systems we're looking at. And this is all offered alongside end-to-end analytical support through Owlstone. So just going to go into the importance of having an accurate and a precise method. It's important for both targeted and untargeted analysis due to their critical roles in reliable quantification, quality control, data interpretation, and cost efficiency. In the targeted analysis, precision ensures consistent and reproducible measurements for specific compounds. Meanwhile, in the targeted analysis, the accuracy and precision are essential for the confident identification of unknown compounds and meaningful pattern recognition within complex data sets. Additionally, these qualities facilitate method validation, detection of low concentrations, and enable reproducibility and comparability across different samples, instruments, and studies, which underpins the validity and usefulness of analytical data. So what can our method do? Well, initially we set up some criteria for how we wanted to validate the method. For accuracy and precision, we wanted plus or minus 20% for 70% of our target compounds, and that's 70% of the 150 that we were validating for. So accuracy here relates to the closeness to the theoretical injected amount, and precision is the repeatability between subsequent measurements injected at the same amount. So for accuracy, more than 75% of our targets passed for the lowest concentration, which is the most critical concentration for us to look at, and upwards of 80 and 90% for the mid and high concentration ranges. For precision, again, we passed with nearly 90% of compounds falling within that variability limit for the lowest concentration, and almost all compounds passing for mid and high concentrations. So these results from our method validation showed that we hit good accuracy and precision thresholds and can therefore stand to benefit from the things mentioned in the previous slide. For example, reliable quantification and then being able to compare between data sets being some of the key ones that we're really interested in. So to summarize what we've talked about some of the applications available through Owlstone. We've focused on the breath biopsy workflow with the breath biopsy collection station and receiver breath sampler, which provide reliable and comprehensive end-to-end platform for breath analysis. This couples with our newest analytical approach, giving validated identification and accurate quantification for a wide range of VOCs. This includes many commonly linked to biological processes, including inflammation, metabolism, and the gut microbiome. And finally, we've talked about the addition of new significant targets into our VOC atlas and how important they are to know what's on breath. So I'll hand back over to Hugh to conclude the talk.
Thanks, Tilly. Yep, so I hope you enjoyed this introduction to Owlstone Medical. We're going to be running more webinars over the course of this year into next year, talking about more specific applications of the technology, but we wanted to just provide an update today on some of the advancements in the technology we've made over the last year or two. As I said, if you have any specific questions or want to find out more after the webinar today, please feel free to email us at breathbiopsy@owlstone.co.uk. And if Tilly, you can just go to the next slide. And just to mention as well, we also run our annual Breath Biopsy conference. It's a virtual event actually since COVID, which we feel works very well. We generally have somewhere between 700 or 800 people joining and presenting research across the breath research community and others. So that's taking place on the 5th and 6th of November of this year. It's online and free to register, and you can get the details of that on our website. So with that, I think we can turn to the Q&A and some of the questions that have come through while we've been speaking today. So first question here, I'm interested to know if your breath analysis testing technology can be used to investigate Parkinson's disease, which is a very interesting question. It's not something that we've explored here at Owlstone. Of course, it's an area that could be applicable to breath research. There's actually a lady from Scotland actually called Joy Milne, who was able to smell Parkinson's disease on her husband 12 years before diagnosis, which is quite an interesting anecdotal point here. But of course, yeah, with the technology we have, we can look at any disease, and we just need to have the right sets of disease and control patient samples to profile the volatiles and see which biomarkers appear. So that would certainly be a very interesting one to look at. Other question here, does Owlstone consider SIBO analysis? So yeah, SIBO or SIBO, small intestinal bacterial overgrowth. As I mentioned during the talk, we do provide that as a service to the National Health Service here in the UK, but we can also make those kits available to others. And yes, they do measure hydrogen and methane gases. Diagnosis of SIBO is related to the appearance of those gases after taking a carbohydrate substrate and the profile that's produced. So, and that's used to make a diagnosis of SIBO. As I mentioned, we're also about to launch a handheld device for measuring hydrogen and methane, which could allow for more longitudinal monitoring at home of those gases as well. Those gases are not measured on our OMNI research platform, though they're too small, but we have other methods to do that. Does Owlstone look at diagnosis for long COVID and related illnesses like ME, CFS, long Lyme? We don't have any active programs in that area, but again, I think that would be a very interesting application to look at. So if Mark, if you have access to such patients, we'd certainly like to talk to you about what we could do in that area. And then a follow-up question from Mark here. Can Owlstone look at exhaled gases such as ammonia, H2S, COS, and nitric oxide? Again, we don't measure those on the OMNI platform, but we do have other ways to measure those gases. Certainly hydrogen sulfide we can measure with a different method, and ammonia. COS and NO, I'd have to check on that. I don't know, Tilly, if you're aware of that at all. Nitric oxide. I know they're too small to measure on the OMNI platform, but
yeah, it would be a different technology.
But yeah, we certainly could measure them using different other methods. Yeah, question here. What would you do for compounds not suited to your current panel? Do you want to take that one, Tilly?
Yeah, so our R&D team are currently in the process of investigating orthogonal methods. So that would be a different column hardware to cover compounds that might not be suited to the current column polarity. So it might be that we end up with a suite of methods that all look at different groups of compounds to kind of broaden the landscape there.
Yeah. Thanks, Tilly. And then there's a question here, interested in the measurement section, kindly can you share what kind of sensors are used in the devices? We're fairly agnostic actually on the type of sensor that we use. We can source those commercially. Within the OMED device, we use metal oxide sensors to measure the hydrogen and methane. But we're constantly looking at different types of sensors that we can incorporate depending on the type of VOC that we want to measure. We do have our own technology, the FAMES technology. So that's field asymmetric ion mobility spectroscopy. So that's a type of IMS that can be reduced to a small format footprint, which is tunable to measure different VOCs and has a high sensitivity and different flexibilities for different types of applications there. So we would either take commercially available sensors, or we'd use our FAMES technology for, depending on the application and the type of volatile that we want to measure. Can breath sampling be done at home? Yes, I think I've already provided an example of where we've done that in the area of gastrointestinal health. But also within our test programs, we're also designing other breath collection methodologies for collecting specific gases on breath related to the work we're doing in lung cancer and liver disease. So the method that we described here is a research method for profiling a very large number of volatiles for biomarker discovery. That does need to be used in a lab or a clinic with a supervision. But once we move into the final test, then yes, it could be developed potentially into home-based type of approach. How amenable is your breath collection to high-frequency serial sampling? I touched on this in the beginning. I think because it's breath, and because it's non-invasive, it's obviously very suitable for serial sampling. Obviously, that becomes more challenging if you've got to take seriously a lot of blood or stool samples, for example. So I think that is one of the benefits of breath. Could you mention the work and progress with tuberculosis? Yeah, so the TB work we're about to initiate, actually. We can't talk about it here today, but we will be putting out a press release on a major effort in tuberculosis over the coming weeks. So I'd say watch that space. We can't talk about it today, but we should be able to announce that very shortly. Are biomonitoring tubes the best for short chain fatty acids in breath? Do you want to take that one, Tilly? Because I know we've been doing some work on that as well.
Yeah, so I think the best answer I can give to this at the minute is we haven't tried different types. I think what we're trying to do is have an overarching method that uses for the vast majority of the 150 compounds. It might be that something may work better. However, we would say that we have obviously validated the short chain fatty acids that are currently within our panel, and we're happy with the results. So the plan for this wasn't necessarily to focus in on those compounds and have a very highly specific method for them. We're quite happy with their performance on biomonitoring tubes on our current method. I think it's the easiest way to answer that one.
Yep. Next one, how do you validate each biomarker? Is there or was there for your Atlas an actual biopsy? Well, we generally will benchmark against any available gold standard that we have when we're running a particular study. So, for example, in our liver disease program. So this is for NASH, now called MASH, and cirrhosis. We would generally be benchmarking against a liver biopsy. If that's not available, then it would be some imaging approach such as FibroScan. But we try to benchmark against whatever gold standard is available to us when we're running these biomarker studies. How do you validate the biomarker? I'm considering whether, and this may become or could be used as a screening method. Yeah, again, I think screening is a very legitimate approach for breath analysis, and for both our lung cancer program and liver disease program that I didn't get a chance to talk about today. We are considering screening applications. So screening generally in high risk populations to triage to, for example, CT scan in the case of the lung cancer program. In case of liver disease, it would be triage to specialist hepatologists, for example. So I think, given that you can run these types of assays in primary care or even in the home, I think it does make them amenable for this type of screening application. But we're in the process of running the clinical trials to prove out the biomarkers before we move to that point. What stage of development for the volatile compounds associated with various tumors? Yeah, as I said, most of our work is being done in lung cancer at the moment for cancer-related biomarkers, and there we are taking a probe-based approach. So this is to take a volatile probe that's metabolized by an enzyme that's in the periphery of the tumor and then releases a volatile reporter basically back on the breath. And I was described it as phase 2 clinical trials at the moment for that. Does SIBO handheld give direct readings at home? Is H2 via electrochemical sensor and it's methane via an NIDIR? Okay, so Mark, it's from Mark. So yes, the handheld is designed to give a direct reading at home onto the mobile phone app. So that is how that works. The hydrogen sulfide is not part of that measurement. That would be a separate approach currently, but at some point in the future we might be able to do that. The methane measurement, I believe, is via an MOS sensor. Great presentation. Do you think machine learning algorithms will be more helpful in analyzing breath metabolomics data, especially when collecting samples longitudinally? Potentially, I do think that if you are using machine learning algorithms, ultimately, I think you still need to identify and validate those biomarkers and understand how they relate to the biology ultimately, rather than just taking a pattern-based approach. I think once you've got a very robust platform of the type we're developing here at Owlstone, it does become possible to use those types of approaches. Tilly, anything to add to that from your perspective, do you think?
No, I think that's covered it quite nicely.
Yeah, so there's a question here about Indigenous population health surveys and the ability to collect samples out in the field. Yeah, we'll probably follow up on that one afterwards. That's quite a complex one to address here. I've already addressed the AI machine learning question. Yeah, so again, this is an interesting one. Working with 3D printers, want to know if volatile affects health and whether this is a good method, and whether 10 minutes of breathing and concentration was, yeah. So generally that breath sample takes about 10 to 15 minutes to collect. And I think, yes, it could potentially be interesting to look at volatiles that are being emitted by 3D printers. So we could look at the levels of those volatiles that are being absorbed on the breath. but we could also look at the effects on the host in terms of, for example, an inflammatory signature coming from the lung, or other aspects related to the impacts of those volatiles on human health as well. So, and we've got other similar types of studies ongoing in different areas around exposure. What are the potential applications and benefits of Owlstone's technology in the field of medical diagnostics, and how does it compare to traditional diagnostic methods? That's a big question. I think for me, this is my personal opinion. I think the main advantage is to be able to have a method which is fieldable or used in the home, which are at relatively low cost ultimately. Rather than having people come into a lab to give blood samples or stool samples or whatever it happens to be. So I think it's just the convenience for the patient and being able to develop ultimately a method that's relatively low cost. I think we've kind of addressed that next question over the course of the presentation in terms of what are the key components of the collection station. Can you measure carbon monoxide? I don't think we've tried to do that. We can't measure that with the OMNI platform, but we could potentially do that with other methods within the business. I don't know about sensitivity on that. I'd have to get back to you on that one. I think that is pretty much all of the questions. So unless there's anything else, we can probably end there. And again, I just say thank you very much for joining the webinar today. Thank you, Tilly, for your presentation as well. And yeah, please get in touch if you have any additional questions or would like to follow up with us using that Breath Biopsy email address. I think we can end it there. Thank you.
Thanks, everyone. Take care. Bye.